And that brings me to something that we're more likely to see a lot this term, a technique called fluorescence in situ hybridization, or FISH. So this is fluorescence in situ hybridization. And if you do it on fish, then it's called fish-fish, which is what I did for my dissertation, essentially. It was a lot of fish-fish. So here's how fluorescence in situ hybridization really works. So you make a metaphase spread, so you have a slide, microscope slide, and you drop cells from your fish that you want to study onto it, and you try to find a nucleus that has metaphase chromosomes. So they all fall somewhere on the slide, and some of them are on top of each other, and some of them are overlapping each other, which is annoying as hell. You have to look at lots and lots and lots and lots and lots of nuclei before you find one that you can actually use to study, because chromosomes never behave. So what's the next step for fluorescence in situ hybridization? Now we've got chromosomes to look at. We want to know, as you suggested, where's the position of a gene that we know is on the X chromosome? Where is it located on the Y? So the way we do that is using bacterial artificial chromosomes, in our case, big circles, abbreviated BACs. Bacterial artificial chromosomes. Huge chromosomes, maybe 200 kilobase pairs in size. Normal plasmids that you might work with in the laboratory are usually like 5 kilobases, 10 kilobases, 7.5. These are way larger than that. And this BAC contains a little bit of vector sequence, the stuff that helps it reproduce inside bacteria. The rest of this, though, the rest of the bacterial artificial chromosome is stickleback genomic DNA that's been purposefully cloned into that vector. Genomic DNA. So you got a little bit of bacterial sequence. The rest of it all came from the three-spine stickleback chromosome. So in this case, let's say it's from the X chromosome. And we know there's a gene in there that we want to know where it's located on the X and the Y. In this case, say isocitrate dehydrogenase, IDH. So we've got chromosomes on a slide, all of the stickleback genome. And we've got one piece of DNA that we've grown up in a test tube and isolated and purified. So we have a tube full of pure chunk of X chromosome containing the isocitrate dehydrogenase gene. So the question is, how do we figure out where this piece of DNA from the back came from in all of those chromosomes on the slide? So, the first thing you have to do is denature the chromosomes that are on that microscope slide. What does it mean to denature?
Separate all the strands.
Yeah, some people also call it melting. You melt the DNA, you denature, you separate the two strands of DNA. So that now what you have on the microscope slide is the two strands of A chromosome with their base pairs hanging out. Not double-stranded anymore. And you've got the other homolog. They're joined together at the in metaphase of mitosis. They're joined everywhere, but there's some sort of cellular structure there that holds the two homologs together. If somebody thinks of it, you can tell me. Otherwise, we'll just leave it. So let's say this is the X chromosome. And let's say that's the region of the X chromosome that our bacterial artificial chromosome insert came from. So then you take the bacterial artificial chromosome and you melt it, denature it as well. Oh, and by the way, this bacterial artificial chromosome has been fluorescently labeled, which I'm going to use asterisks, and it's nice that I have this bright fluorescent color to illustrate this. So chemicals have been used to modify that solution of purified DNA we had in that test tube that contains part of chromosome X. So now we've denatured it. Now what? It's the H in fish. We've done the fluorescence. Yeah, we've done the fluorescence part. We've labeled the probe. The piece of DNA we want to know, where does it go in the genome. The in situ part is we're going to find out where it's located. And then the H, hybridization. So we hybridize this poor single-stranded probe to those chromosomes that are laying there on the glass slide with all their base pairs hanging out, waiting for a base to bond with. So what that winds up looking is you get X chromosome, and wherever that probe is from will hybridize by typical ATGC base pairing to the part of the chromosome that it came from, the homologous part of the chromosome.
That'd be like Taq polymerase usage, or?
Oh, sorry, did I say?
No, I was just asking, like, how would you, how they bind them?
Oh, just by thermodynamics. Yeah. So you put it in the right buffer so that base pairing is possible, and that that single-stranded DNA anneals specifically to the place in the genome where it is most similar. I could talk for days about that, but I certainly will not. Okay. So you're looking at a slide, you see two structures crossing each other, the two different homologs of the chromosome, and on each of those, you see two fluorescent dots. Each one of those dots is one strand of the chromosome bound to one strand of the probe. And the probe is fluorescent, so it's shooting out light. You're seeing a microscope. You're seeing this beautiful light and thinking, holy crap, this experiment actually worked. And this is what you see. And if I turn down the lights, it'll be even cooler, maybe. If I can turn off the lights. Maybe not. Can you guys see that well enough? I don't know. Okay, so what are we looking at? This is an entire metaphase chromosome spread. All 21 pairs of chromosomes, plus this blob here. That's not a chromosome. And I've hybridized, in this case, oh, it was IDH, good for me. A piece of X chromosome containing the isocitrate dehydrogenase gene, which is labeled in green. So those molecules are fluorescing green. Can you see where they are? There's a dot right there and a dot right there. Both homologs of the X chromosome. And then a pair of green signals right up there. Of course, the two things are labeled X chromosomes. So what does that tell us? The technique works. The isocitrate dehydrogenase gene is somewhere on the middle of the long arm of the X chromosome. Okay. So this is from a female, 2 X chromosomes. Are they homomorphic or heteromorphic? If you cut them out and spun the one upside down and lined them up, they're homomorphic. Can't see any differences. Not only are the chromosomes the same relative length, if again you cut that one out, flipped it over and moved it next to the other one, same relative length, same shape, centromere is about in the same place, sort of off towards the top, and the IDH probe, the position of that gene, is about the same, as far as we can tell, position on the long arm of the chromosome. So 3 different pieces of information: overall length, location of the centromere, and location of any fluorescent probes that we've applied to the chromosomes.